So starting from the left, the company actually is quite integrated for a European biotech company. That means we have our discovery platform that I will explain to you in a minute a little bit more in detail. But besides antibody discovery and target discovery, we also do our own manufacturing. So we have the capability to go up to 1,000 liter GMP, can generate our own cell lines. We can even do our own formulations. You will later see that one of our assets is an IV, as antibodies normally are, but the other one is a sub-Q formulation that we have generated in-house. And this mix of capabilities gives us really a very strong competitive advantage. And besides building our own portfolio, also creates licensing and partnering opportunities. In our pipeline, we are currently focusing, and we just went through a prioritization a couple of months ago on our two lead programs. One is called BI-1808 and the other one BI-1206, which are both first in class with a differentiated mechanism of action in areas of high unmet need and more importantly, multiple upcoming value inflection points, as I will tell you later. So both assets actually, as I already said, are first in class and are active in liquid as well as in solid cancer. And I will give you later some more details there. We have done already a number of partnerships based on our platform. We have done more than 10 technology validating deals with the companies you see there: Pfizer, Daiichi, Bayer, Mitsubishi, Takeda, Genentech. And on top of that, we have strategic partnerships with Transgene, Merck Sharp and Dohme, AstraZeneca, and Kasi Pharmaceuticals. With Kasi, we have done a deal for China, including Hong Kong, Macau, and Taiwan. And just recently, also just a few months ago, we sold one of our non-core assets to Soma, where we would have received, in some time, quite decent royalty to Soma, but we thought cash now is much better than maybe a little bit of royalty in a couple of years, and that generated another 30 million US. And with this, we're actually well funded. That means we have a runway currently based on current plans into Q1 2027, and that will cover multiple upcoming near-term catalysts, as I will tell you in a minute. Before I go to the programs, as I said at the beginning, so I wanted to introduce you very briefly to our discovery platform, which is called FIRST. And the reason why I want to do this, because this is really unique for Bioinvent. So this is a platform which allows us to screen directly on patient material. And then once we have identified interesting antibodies to the tumor materials from patients, before we then identify the target, we characterize the function. So that means we test it in vitro and in vivo in animal models. And when we see strong therapeutic effects in animals, then we identify the target. So this is basically a so-called reverse phenotypic functional screening. Very complicated, but what it actually does, it allows us to identify new immuno-oncology targets, as we have done for our two lead programs, TNF receptor 2 and Fc gamma R2b. And also we can identify new uniquely functional epitopes on already validated targets. So we have, for instance, identified an anti-CTLA-4 antibody that seems to be actually more potent as the current one that is used in the clinic, ipilimumab. And the reason for that is that it binds to a different functional epitope compared to ipilimumab. So those are the two advantages that we have with this platform. And obviously most important are those novel immuno-oncology targets, as I will tell you. So on this slide, you will see now our current portfolio. So starting from the top, this is BI-1808. This is a program targeting TNF receptor 2, and I will tell you later a little bit about the target. And we developed this in solid tumors as well as in T-cell lymphomas. And we run it currently in clinical study as a single agent, but also in combination with pembrolizumab. And that is based on our preclinical assessment, where we saw strong single agent activity, but also very strong synergies with pembrolizumab, which is Keytruda, which is the top-selling oncology drug from MSD, Merck. And as you can see on the right-hand side, so pembrolizumab, we don't have to buy. We get it for free from Merck because we do this under a supply and collaboration agreement with Merck, which means Merck does not have any rights, but can contribute to the clinical development. And of course, for us as a small biotech company, this is interesting because then we have a potential partner already at the table. So then the second program is BI-1206, which targets Fc gamma 2b, another very interesting immuno-oncology target, which I will explain to you also a little bit more details later. And this we develop in non-Hodgkin lymphoma and in solid tumors. And in non-Hodgkin lymphoma, we already in phase 2. In solid tumors, we just started the phase 2 actually in combination with pembrolizumab in first-line non-small cell lung cancer and uveal melanoma. And you see here again Merck, where we combine it with pembrolizumab. And you see also here AstraZeneca, because in non-Hodgkin lymphoma, we combine it with rituximab and acalabrutinib, which is one of the important compounds of AstraZeneca. And it's basically the same arrangement that we have with Merck. So it's a supply and collaboration agreement. And then you see also CASI here, and CASI is our exclusive partner for 1206 in China, Hong Kong, Macau, and Taiwan. So what I will do now is go a little bit more into detail of the programs, and I will start with our anti-TNF receptor 2 program. I introduced already the antibody 1808. And TNF receptor 2 actually we discovered obviously with our first platform that I briefly introduced you at the beginning of my presentation. And we found it an interesting target in the tumor microenvironment of solid cancers, but also as a target on T-cell lymphomas because it's overexpressed there. So that's why we can develop this in T-cell lymphoma and in solid tumors. And here you see a little bit of the positioning of 1808. I start on the left-hand side. So CTCL, it's a rare T-cell lymphoma. We have already there very, very excellent data that we published during the summer with 100% disease control, a very high overall response. Data is still maturing, a very good safety profile. That's also something that we're really focused on because you not only want to have a product or a drug that is efficacious, but also very safe. And in that sense, we believe it's positioned to become the frontline monotherapy treatment of choice based on the data that we have seen so far, because all the current therapies are limited by safety and efficacy. So that represents for us a very attractive near-term market opportunity of roughly $500 million US, based on very bad drugs that are currently on the market. Since we have a good safety profile, we think we can go in earlier stages of the disease, which means that could be even more than 500 million US. So then on the right-hand side, solid tumors, which is of course the larger commercial opportunity. There we're talking billions. There we have shown, and that data is already know, single agent activity and anti-tumor immunity across various malignancies. Ovarian cancer, we had a very strong complete response. We saw a nice response in non-small cell lung cancer, but also in GIST, which is a very difficult to treat tumor, where we saw also a strong, robust partial response that might be already now a complete response. So really promising signs of efficacy. Again, also very, very favorable safety profile. And those data that I mentioned here for CTCL as well as for solid tumor is, I'm talking single agent. So this is single agent activity, which is actually quite important and very exceptional. So going a little bit into the data that we had with CTCL, that was a poster that we showed at EHA this year. I mentioned already we had 100% disease control in 9 evaluable patients at that time point. You can see the breakdown here. So there was 1 complete response in Sézary syndrome, which is a very late stage form of CTCL. Then we had 3 partial responses, and then a number of patients with stable disease. And it goes without saying, all patients that we're treating here have seen the standard of care and no other treatment options anymore. So I think the data that you then see here is very strong data. And then we also had 2 responses in PTCL, which is even a more severe form of CTCL, where we had 1 partial response and 1 patient with stable disease. And in addition, as I already mentioned, it's super well tolerated. We had just mild to moderate adverse events, just grade 1 to 2. And we could also immune activation, could observe immune activation early on with depletion of the regulatory T cells and an influx of CD8 positive T cells into the skin. And that's what you want to see when the immune system is reacting towards the cancer. We have, as mentioned here on the right-hand side, orphan drug designation for T-cell lymphoma and fast-track designation for CTCL. So based then on the data, so the potential path to first approval looks like this. So we have orphan drug designation, we have fast-track designation, and we have finished the phase 2 part A, as you can see. And currently, we are also running this in combo with pembrolizumab, just to see how it looks in combination with pembro. As I said, I'll just repeat all the data that I've shown you so far as single agent, which is very, very good. And then in parallel, we'll start with dose optimization such that we then hopefully next year can get breakthrough designation, agree the phase 2 design, and then hopefully start a potential pivotal study in 2027. So that's at least the plan. And what we're doing is we're basically planning it as we would do it on our own, but we are also, and we come back maybe later during our Q&A, looking into quite heavily into partnering. So this could be also done with a partner, and that would be also my current preference. Then going to the solid cancer side from 1808. So I mentioned that we saw, and that was presented at ASCO in 2024, very interesting signals in ovarian cancer, in GIST, and in non-small cell lung cancer. So really a nice single agent response profile. And then based on that, we went into the combination because, as I mentioned, we saw single agent activity, but also strong synergy with pembrolizumab, with Keytruda. And at the end, especially when you look at solid cancer, so it's good that we have single agent activity, but at the end, if you really want to treat solid cancer in a good way, you need combination. That's why that's also something that you need to test in any way regarding then efficacy and safety. And we will have already an update for the solid tumors in combination with pembrolizumab this year. So it's coming towards the end of this year. Okay, now I'm switching to the other target, anti-Fc-gamma2b. Fc-gamma2b is a very interesting target for two reasons. It's number one, expressed on tumoral B cells, where it's part of the resistance mechanism to anti-CD20 rituximab. That's why we develop it in non-Hodgkin's lymphoma. But it also is expressed in the solid tumor environment on a very important cell type, the dendritic cells, and it's the only inhibitory receptor on dendritic cells. And when we block it, we take away this inhibitory signal. That's why we develop this also in solid tumors. Here again, the positioning. So for non-Hodgkin lymphoma, I think we have strong second-line potential with a triplet combination, as I will show you in a minute. We are on track for overall response rate of 75%. This is a chemotherapy-free regimen, I think, which is very important because non-Hodgkin lymphoma patients are normally elderly, frail patients. And this antibody is actually available in the sub-Q formulation, and that, of course, improves the convenience together with the oral acalabrutinib and adds additional flexibility. We saw a very good safety profile compared to our competing drugs, no cytokine release syndromes, no neurotoxicity, which supports a broad use, including in community hospitals, because that's what people want. And anti-CD20, even though rituximab is a very old antibody, probably the first one that was introduced to the market, will remain core and center for the treatment of non-Hodgkin lymphoma for at least next 10 years. So I think from that sense, from that perspective, we're very well positioned. Then on the other side, solid tumors here as well, which of course is the largest commercial opportunity. And there we have the next trial in first-line lung cancer. So that has already been started, actually. We announced the activity of pembrolizumab and are really focusing on patients that do not respond anymore to anti-PD-1 or anti-PD-L1. And we saw very strong signals in heavily pretreated patients with metastatic melanoma, cutaneous melanoma, as well as uveal melanoma, very difficult to treat. And we think we can extend that to other tumor types. So it's ideal for a combination component with anti-PD-1, anti-PD-L1 in several tumor types. Looking a little bit into the data, because data is always our guidance. So this is, again, in non-Hodgkin lymphoma combined with rituximab, as you can see. a very good overall response rate with a high number of complete responses. We had 8 complete responses out of 22 patients, 5 partial responses, and 6 patients with stable disease. And I think what is very important here is that the complete response have a long duration. So we have now a number of complete responses that are long-lasting, and 3 of them several years after the end of treatment. That's exactly what you want to see, that you have a high-quality, long-enduring response, and that's what we're seeing. We wanted to kick this a little bit higher, the objective response. That's why we combined it also with acalabrutinib, and this data we have shown earlier this year. And we had a preliminary objective response here of 63%, but I think I can tell you that we are very much on target to achieve one higher than 75%. And we saw also that this treatment was very well tolerated. So out of 8 patients that we have, out of 30 patients that we will have at the end, we had 2 complete responses, 3 partial responses, and 3 stable disease, which is 100% disease control. So this looks very, very promising. And the next update will be during early next year, where we then hope to have the complete 30 patients. And as I said, so we're on target for objective response higher than 75%. We have orphan drug designation for follicular lymphoma and mantle cell lymphoma. And so this is a very interesting study to follow. Based on this, we have also made some planning how we could go about it. So the phase 2A signal seeking will be done during early next year. We are starting to planning for a pivotal study, but this study definitely we will do only in partnership with a larger company. But already next year, we can hopefully agree on the phase 2 design and the breakthrough therapy designation, and then could do a pivotal study already at some time point next year. But again, this is something that we're seeking to do under partnership. And as you might know, we are already having a supply and collaboration agreement with AstraZeneca. So that could be one natural party to discuss with, but we also have other opportunities and possibilities. Then quickly at the end, the solid tumor case for 1206. So basically that's what we saw. So those were patients that have been heavily pretreated, at least 2 in both cases where we saw response, even 3 cases, 3 regimens of IO-containing treatments. And we saw in those patients really long-lasting complete responses and partial responses and a number of stable diseases. We showed the data to Merck. And Merck was intrigued that they agreed to start with us a phase 2A in frontline non-small cell lung cancer and uveal melanoma. And you might have seen the press release a week ago or 2 weeks ago. So that has already been started. And that data then should be available, first glimpse of the data, at least by second half of next year. So very, very interesting. And this is also something where we feel if that works, that can be then extended into other indication in addition to non-small cell lung cancer and uveal melanoma. And keep in mind, 1206 is sub-Q. You might have seen that the sub-Q version of Keytruda has been approved. And if you only would increase, just to give some numbers here, the response of Keytruda by 10%, I think this is very, very interesting for Merck. And you could easily then think to combine 2 sub-Q formulations in one application. And that could be a very, very interesting and powerful treatment. So at the end, this is just a summary, and I think I don't go through this again. So those are the best clinical responses that we have seen in both programs. As you see, quite a number of patients that are responding, including complete and partial responses. And then I will end with the key catalysts for this and next year. So during this year, there will be then an update on 1808 in solid tumors. That will be phase 2A data in combination with pembrolizumab/Keytruda. And then first half of next year, we'll have further T-cell lymphoma data, this time in combination with Pembro. And then also the triplet data for BI-1206 that I mentioned, the 30 patients should be available as well. And then last but not least, very interesting, also the first readout for the phase 2A data with pembrolizumab in first-line non-small cell lung cancer and uveal melanoma. Thank you very much.
Thank you very much, Martin, for an excellent presentation. We have lots of questions from the audience. Let's start with Fredrik. He wonders if it's always clear for you to develop the asset into phase 2, or have you had the opportunities to partner with pharma based on phase 1 data, but opted to take it further on your own?
Yeah, so we did one partnership with CASI Pharmaceuticals, as I mentioned, that was based on phase 1 data, because they already had a nice profile when we just combined with, now I'm referring to 1206 with rituximab. And then since we raised quite some money, we thought it would be better to go one step further into phase 2. And then nowadays, in any way, I think what is needed in order to do a good partnering, you need phase 2 data. You can't do it on phase 1 or even preclinically. So that's not possible anymore. And of course, also you would get a higher value at that time point.
Yeah. Another question regards the share price. I'm not really a fan of companies discussing their own share price, but let's rephrase it and say, can you mention what are the most common questions you get from investors about value creation with Bioinvent? What are the key areas that they're looking for or questioning?
Yeah, so basically, the number one question is when are we in a position to do a partnering? That's a very I would say number one priority questions, and I think a very legitimate one. And regarding this, I can say that we are now in a zone where we can do it. So we have several discussions, and I think also the supply and collaboration agreements that we have with Merck and AstraZeneca, I think, is a first step. So like this, you already have two companies, big pharma, that are at the table for potential partnering. But of course, in the back, we have additional discussions, even already under CDA. So I think if the data continues to develop like it does at the moment, I would think first half of next year should be a very interesting year regarding partnering.
Mm. With regards to HMI-115, you recently delivered strong clinical data there. So what's the prospects of monetizing this asset in the near term? Could you speak a little bit how you want to maximize the value of that?
Which asset you are referring to?
HMI-115, 115.
HMI-115. Is that the compound?
The Hope.
The compound that we sold to Soma? Is that?
Yeah, I guess that's the question. Yeah. So let's, you already answered that. And another question is, sorry, I should screen this a little bit before. Perhaps you could elaborate a bit on the rationale behind the recent decision to test BI-1808 in combination with pembrolizumab in CTCL.
Yeah, so the reason for that is that we just want to see how it looks. So obviously we have very strong single agent data that would be already enough and sufficient to go for first line, as I already explained during my presentation. But nevertheless, I think it's good to see how it looks with pembro, because it might be even better.
Okay.
And that's why we do it. We don't have any preclinical information that would guide us into this direction, but we just thought, we can include a couple of patients just to basically rule it out. Is it as good or maybe even better? And if it's even better, then of course, then you would go for combination therapy, and that could be also something very interesting for Merck to look at. Mm. And regarding the other question, now I might think what it could be, because the Takeda asset obviously has been sold. Maybe this is the one that we have with Bayer Hope, because there's another non-core asset that is now transitioning soon from phase 2 to phase 3. And that's indeed something that we're also looking into to monetize in case somebody is interested, absolutely. But more I can't say at the moment, because all those non-core assets that might generate at some time point royalties, which are normally lower royalties, is not so sexy, because all those deals were done very, very early, where you only would get a low royalty at that time point. So I think for us to monetize it now is much, much better than to monetize it or to wait for the royalty stream. So I think that was probably the question, because the Takeda asset is gone. But there's another one that is now moving from phase 2 to phase 3 soon, and that is with Bayer Hope. Absolutely. It's something that we're doing. Yeah.
That was interesting to hear about BA-1808 too, because it seems to be a bit of a change in direction from what you stated this spring. And in the preclinical portfolio, are there any assets there that you have more hopes on in relation to the others? What do you think is the key assets in that part of the portfolio?
So for us in the portfolio, so we went just recently through this strategic evaluation. So those two assets, 1206, 1808, are by far the most interesting ones. It's I always say it's an unfair competition, because obviously they're also the most advanced, where we have clinical data, we have safety data. With the other programs, we don't have that, right?
Yeah.
But at some time point, you have to double down, and that's what we did. But we also still have preclinical work that we do, not only in immuno-oncology, also in autoimmune. We're looking at that, but we would then inform the market once we feel we have a clinical development candidate, which we don't have yet, but maybe soon. Maybe soon. Yeah.
I was looking earlier at some M&A data for Europe for the third quarter, and it seems okay. I mean, it's not really
it's picking up.
Fantastic. Yeah. So what's your feeling for the M&A market currently?
As I just was saying, so it's picking up, and also not only in Europe, but also in the US, definitely. And that actually has helped for the most recent financing. So there were a couple of larger ones also in Europe. There was a German company that I think received a 300 million financial injection. And that is only possible because there was a return of capital through M&A, right? So M&A is definitely picking up, and all the people that I talk to, whether it's banks or analysts or other people that are in the field, they say clearly M&A is picking up, and that's also my field. So we have a lot of discussion with pharma, potentially interested pharma companies. They're actually following us. They're reaching out to us, but obviously what you would need is more mature data. But we have those discussions already now, and as I said already earlier, I think first half of next year should be a very interesting year in this respect.
Well, you presented the timeline, which shows the readouts. So we have a lot to look forward to. That's all the time we have tonight, unfortunately. Thank you very much, Martin, for participating tonight.
Yes.
And good luck with your research.
Thank you very much, Lars. Thank you.
Thank you.
Until next time. Bye-bye.
Bye. So it's time to move on with our schedule. It was very interesting to hear Martin talk, as well as the other companies.